Okay, hi everybody. So today we're going to be talking about the neuromuscular junction and basically synapses in general. So let's take a look at what we already know and what we've already covered. So if you recall, we drew our cell bodies, right? Or we drew our nerve, and we had our dendrites at one end, which received communication. Then we had the cell body, and then we had the axon, which would take basically electrical impulses or signals away from the cell body. So what we said is, if we draw this graph again, we said that the inside of a neuron is at about negative 70 millivolts. And then what would happen is we would get stimuli and things like that, and it would cause it to come up to negative 55 millivolts. And this, we said this was called threshold. When we got to negative 55 millivolts, that's when we would have an action potential. So let's draw this real quick. And now we get some type of stimuli. We came up to our negative 55. And what would happen at negative 55 now is these sodium gates here would open. And sodium would start rushing into the cell. Right? I get a whole bunch of sodium rushing into the cell. Well, because sodium has a positive charge, what happens is this starts to become even more positive because I'm adding a bunch of positive charges into the inside of my nerve. So this would go up to, then we said what would happen is this goes up to about plus 30 millivolts. Right? And we said when it was at plus 30, what would happen is these sodium gates would close, and now my potassium gates open. So here's my potassium gates open, and here comes my potassium rushing out. So now potassium rushes out. And then what we said happens now is because potassium's rushing out and potassium's positively charged, this starts to go down negative again, and then it goes to hyperpolarization, cuts back to the rest of the membrane potential, and this closes. And then if you recall, we said we had our refractory period in this time here. The next thing we said that would happen is the next sodium gate would open and close, and then the next potassium gate would open and close, and the next sodium gate opens and closes, the next potassium gate opens and closes, and so on down the axon. So what we're going to talk about today is what happens when we get to the end here. And at the end of the axon, you have enlargements called the synaptic knob. Now, different books call these different things. I've seen it called the presynaptic terminal. I've seen it called the synaptic bulb. I'm going to call it the synaptic knob. The synaptic knob is going to form a connection with three different things, or it can do three different things. It'll only do one of three different things. When it forms a connection with something else, that's called a synapse, and we'll talk more about that in a minute. So a nerve can only synapse with another nerve, right? So my synaptic knob can only form a synapse with another nerve, with a muscle, which is what we're going to be talking about today, or with a gland. Okay? But before we start talking about muscles, let's talk about the connection between axons and other nerves. So I'm going to erase this real fast because I'm going to draw these kind of small. Okay? So now, let's say we have a nerve, okay, and there's a dendrite sticking out, and there's a dendrite sticking out. Here's the axon, and then here's the end of the axon, okay? This is my cell body right here, that's the nucleus. Now, a nerve, like we said, this nerve can do several different things. It can form a synapse. There's my synaptic knob, there's my axon. It can form a synapse with the dendrite of this nerve. Another thing it could do is the axon. Now it's going to look like I have more than one axon, but the axon can also form a synapse right with the cell body of another nerve. Okay, or it can form a synapse somewhere on the axon of another nerve. So even though we say it just can connect to a nerve, there's 3 different areas on the nerve that a nerve can form a synapse with. Now let's look at the neuromuscular junction. So once again, I'm going to draw, here's my nerve, there's my dendrites, here's my axon, and now this is my synaptic knob. Okay, a minute ago I drew these as the little black circles, now this is the synaptic knob. This here is going to be the membrane of a muscle. Okay, and so now what we're going to do is we are going to draw my sodium potassium gates on here. There's sodium, there's potassium. This is obviously an unmyelinated fiber. So here's what's going to happen is, as we saw, and again, this goes all the way down to here. This is going to open and close. 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 This is going to open just like it has before. And like we said on our graph that we drew a minute ago, okay, the inside of this is -70. And now we get our stimulus. This is going to go up to +30. So when this gate opens here, just like it's been doing all along the nerve, when that gate opens there, I'm going to have sodium start rushing in. Okay, and there goes sodium. Now it's rushing into the inside there. Just like we've seen everywhere else, once this gets inside here, the inside of this goes to +30. It goes to +30 millivolts. One difference that we, on the synaptic knob, that we didn't see on the axon, is we are going to have calcium gates. Okay, so over here we have calcium gates. Right there. So here's what's going to happen now. Is on the inside of our synaptic knob, we have something called vesicles. Vesicles are made up of the cell membrane of whatever cell it is. In this case, it's the synaptic knob, but they're made up of the cell membrane. So the way it works is like this. Let's say we have something we want to bring into the cell. Now, a cell can actually reach around and grab it and pull it in, but let's just say this comes in to the cell, and when it goes to come in, what it will actually do is it will push the cell membrane out, come into the cell membrane, and then the cell membrane pinches off, and this substance is now inside the cell. Okay, so now it's inside the cell. I'm not going to draw the red because I'm going to actually draw this real big in a second. This is called a vesicle. This is called the vesicle. Okay, where the cell membrane is with the thing inside of it, whatever that be, is a vesicle. I'm going to give you an example right now. Because we're dealing with muscle and the synaptic knob, and you have hundreds, possibly even thousands of these, of vesicles, but I'm just going to draw one. We have a vesicle on the inside, and the vesicle contains a neurotransmitter called acetylcholine. So the vesicle contains a neurotransmitter called acetylcholine. Remember, this part here is black because it's made up of the cell membrane. That's why I left it like that, and it's going to be important in a minute. On the vesicle and on the membrane, you have proteins. Here's a protein, and here's a protein. Okay? These proteins here are called snare proteins. Now, this is actually snare protein V, which you don't need to know, but this is snare protein B because it's on the vesicle. This is snare protein T, I think it's for transmission because it's going to bring something across. The way a synapse works now is this vesicle, when needed, will now fuse along the membrane again, and it will kick the acetylcholine out, which will cross this space. This space right here is called the synaptic cleft. Okay, that's called the synaptic cleft. Let me write it down here because it's not. Okay, this is our synaptic cleft right here. Okay, now that's the opening between the synaptic knob and whatever this is. In this case, it's a muscle membrane, but the synaptic cleft is an opening between a synaptic knob and either the nerve, a muscle, or a gland. Is the synaptic cleft. So once again, we want this acetylcholine to come across the synaptic cleft and get on this side, so therefore that impulse can continue on. Now, how does it do it? So once again, this is a snare protein too. Okay, both of these are snare proteins. We need to connect those. How does that happen? Like this. This is positive 30 millivolts. And as we know, at positive 30 millivolts, my sodium gates will close. However, just like we saw before, when we have positive 30, we saw the potassium gates open, which they'll do in here too. I just don't have them drawn. But at positive 30, we set the potassium gates open. And again, if on this, I just don't have them drawn, but you have these gates going all around here also. But what happens on the synaptic knob is my calcium gates open. So now the calcium gates open and calcium comes rushing in. Calcium comes rushing in. And here comes calcium. Now, here's the role of calcium. Calcium's going to come between these snare proteins here and form a bond. Okay? When it does that, now the snare proteins will pull this vesicle to the membrane. So I'm going to draw it right here. Even though it would be taking place right here, I'm going to just draw it over here. And this is what happens now. So I have my proteins, right? And now I have my vesicle, and here's my acetylcholine, right? Here's my acetylcholine. Now the acetylcholine can go out into the synaptic cleft, which remember, this is just a space between the synaptic knob and either a gland, a muscle, or a nerve. On this side, we now have other gates. Here's a gate, here's a gate, and here's a gate, okay? And I know these look like the sodium-potassium gates, but they're not. But they will have sodium-potassium passing through them. So here's what's going to happen, is I am going to get, when the on these gates now, we have receptors for acetylcholine. I drew 3 of these. Imagine these are all along the cell membrane, the muscle membrane, okay, called the sarcolemma, by the way. Here comes the acetylcholine. It's going to come out and it's going to bond onto these receptors. It's going to bond onto those receptors. Now, when it bonds onto those receptors, what will happen is these gates open, but I don't have sodium and potassium rushing in. What happens is I'll have 3 sodium go in and 2 potassium come out. See that? So now I got 3 sodium going in, and I have 2 potassium coming out. Okay? So looks familiar. Looks like the other leak channels we had, right? When the cell's at rest. The difference is these closed. The other ones that we saw is when it's just at rest, and those are just like openings that the sodium potassium leak in and out all the time. These open up due to the acetylcholine. So here I'll have 3 sodium go in. I'll have 2 potassium go out. And then I'll have 3 sodium go in and 2 potassium come out. Now, here's the thing, is if you notice, I have 1 more positive charge going in than positive charge coming out. I have 3 sodium going in and 2 potassium coming out. These charges start to add up. These charges start to add up. Eventually, because they add up, we actually call this a graded potential. Eventually what's going to happen is these are going to add up enough that the inside of this membrane here goes to negative 55 millivolts. So the inside of this membrane is going to go to negative 55 millivolts. What happens at negative 55 millivolts? I get sodium potassium gates open and closing. Oh, and look, here's my sodium potassium gates now. Okay, even though I'm drawing everything the same color, here's my sodium potassium gate. So now we're at 55, we're at negative 55. Now this is going to open up. Sodium is going to start rushing in, right? You get a whole bunch of sodium rushing in, right? Until it gets up to positive 30 again. Then these will close, these will open, and now I get a whole bunch of potassium rushing out. And that's going to continue down the nerve membrane, the sarcolemma, until we get to something called the T-tubule, which we'll learn about when we get to the muscular system. So let's go back to this again. I had my impulse coming down, my action potentials coming down. These are opening and closing, 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 opening and closing. Again, I have sodium gates and potassium gates all over this. I just stopped right here. Sodium rushes in, this goes to plus 30. That causes the calcium gates to open, which now calcium rushes in. Calcium forms a bond between my snare proteins. When it forms this bond between the snare proteins, I can now pull this acetylcholine out. And what happens when I pull this acetylcholine out, like you're seeing here, the acetylcholine will come across and attach to receptors on these proteins here, these channels here. Let me draw these a little bit bigger. Okay, onto these channels here. And what will happen now is that 3 sodium go in and 2 potassium go out. But because I have more positive charges coming in than going out, what eventually is going to happen is this is going to go up to negative 55. And then when that happens, now my sodium gates open. When the sodium gates open, then what happens is sodium rushes in, this closes, then the potassium gate opens, that rushes out, and this continues down to the T-tubule. So that's basically it for the cell, the neuromuscular junction. Oh, by the way, what can happen too is you have enzymes that can now go back and break down this acetylcholine, and some of it can go back into the synaptic knob. So that's it basically for the neuromuscular junction. Thank you for watching.